In this video, we are going to start talking about the second class of biomolecules known as amino acids or proteins. So understand that the definition of a protein is an amino acid polymer. What this means is that a protein is made out of many units of amino acids, okay? The term amino acids refers to these basic units used in proteins that contain. If I zoom in, an amine, and that's the amino portion, and then the acid is because they contain carboxylic acid. And each amino acid has a carboxylic acid side and an amino side. And as you can see, they are linked to one another. Now, amino acids are known as alpha amino acids because the amino group and the carboxyl group are separated by one carbon atom, designated as the alpha carbon. So understand that every single one of the amino acids is going to have the amino group, the carboxylic acid group, the alpha carbon, and then they're going to have an R group. So understand that R group specifically is what is different for each amino acid. Okay, so we have to date 20 amino acids that are common to many of the organisms that we know on Earth. Now, all of the alpha amino acids are chiral except one, which is glycine, because in the case of glycine, the R group present in the molecule is another hydrogen. That's why it is not chiral. So most amino acids have one. There are some that have several chiral centers in it, except glycine that has none. One of the things that we need to understand is that amino acids, when they're linked to one another, they can form what is called a peptide. And a peptide is a relatively short amino acid chain. Understand that a peptide, you need a minimum of 2 amino acids coming together, and as long as it is less than 40, most of the time in biochemistry we always think of them as being less than 50. That is considered a peptide. So if you have 2 units of amino acids, that is known as a dipeptide. 3 units of amino acids, tripeptides, so on and so forth. So the prefix di, tri, tetra, penta, so on and so forth, describes the number of amino acids there. Now, understand that a polypeptide refers to a chain of fewer than 40 to 50. Because the thing is that once things get into above 50 amino acid units, then we consider them a protein. What makes these amino acids be linked to one another in these peptides is a peptide bond, which, when we look at it at the chemical level, these peptide bonds are amide linkages between the amino acids. Okay, so the larger the chain, as I just mentioned, so this has to be at least larger than 50 amino acids. Then that molecule is considered as a protein, and it implies that it has a biological function. Now, The 20 alpha amino acids occur naturally, okay, and they differ from that R group that I mentioned a few moments ago. Natural amino acids are called L amino acids, which is different from the carbohydrates because most of the naturally occurring carbohydrates are D. Okay, so when we look at the different amino acids, understand that similar to what we mentioned in the carbohydrates, the L and D configurations for amino acids refer to the RNS configuration that they can have. So understand that for an amino acid, specifically, as long as you have the carboxylate on the north side of your amino acid, the R group or side chain on the bottom, and in the middle, you only have the amino and the hydrogen. If the amino is on the left, then we have an L amino acid. If you have that NH2 on the right, then that's going to be a D amino acid. But again, the most of the naturally occurring amino acids are L. So that's why you see here L-alanine and L-serine. If you are a student that as part of your diet regimen takes amino acids, you will see that the bottle will say L and then the name of the amino acid. When it comes to the amino acids, they can actually be grouped by the polarity of their side chains. So here we have the amino acids that are known as non-polar side chains. So when we look at these and we look among the structures that are present in many of them, one of the things that we can notice is that these amino acids that have non-polar side chains, the side chains are going to have specifically hydrocarbons in them. So for example, alanine has a methyl, valine has an isopropyl. Oh. We have leucine, isoleucine, phenylalanine, proline. All of those are going to have hydrocarbon side chains and they are non-polar. Now, the rest that are non-polar, specifically here, tryptophan is the only one that is going to be a heterocyclic amine. But understand that this heterocyclic amine, it is considered non-polar because the lone pair in the nitrogen is not basic, meaning that it cannot act as a base. Understand that the amino acids also have a 3-letter code and a 1-letter code. Now, if we continue to observe what else we have in the nonpolar, we have glycine. And in terms of the glycine, it's going to have a hydrogen. So that is going to be special among the different amino acids. And again, the other one is tryptophan, but I already explained what is what makes tryptophan special. Now, there are amino acids that have a polar side chain, and those include threonine, tyrosine, cysteine, serine, glutamine, and asparagine. If we look at their structures, specifically for threonine, tyrosine, and serine, we see that they have hydroxyl groups. Cysteine, on the other hand, has a thiol group. Asparagine and glutamine are going to have amides in their structure. And that's what constitutes the polar side chain that they have in them. Let me mention something, because here they put valine 2 times. Let me just go back for a minute. When it comes to the amino acids, the other nonpolar amino acid that is not listed here is methionine. It's just that we see valine 2 times. Methionine is also a nonpolar amino acid. MET is the 3-letter code. M is the 1-letter code. And understand that when it comes to methionine, look up the structure. But you will notice that the side chain for methionine is going to be a thioether, because you're going to have a sulfur in between 2 carbon atoms. But I was surprised that they didn't have it in the slides. So I just want to mention that is the other amino acid that is not listed. Let me move on to talk about the last 2 classifications for amino acids, which are the acidic side chains and the basic side chains. So when it comes to the acidic side chains, as you can see, you have aspartic acid and glutamic acid, which at times you can see them being called aspartate or glutamate. And that aspartate or glutamate just represents the carboxylate instead of being the carboxylic acid. When it comes to the basic side chains, understand that we're going to have lysine with an amine, histidine with a heterocyclic amine. It's just that in this heterocyclic amine, understand that this nitrogen is basic, so it can get protonated. And when it comes to arginine, this specifically has a functional group that we haven't seen before that is called the guanidinium group. And in that molecule, understand that this nitrogen atom that is just double bonded to oxygen can act as a base. They're weak bases. We already saw in the previous chapter how amines are basics. So each one of these part of the molecules that I circle is what acts as a base in any of these basic side chains. When it comes to the amino acids, understand that our bodies can make 10 out of the 20 amino acids. So we need to get the other 10 amino acids from our diets. Any amino acid that needs to be obtained from our diet, it is known as an essential amino acid. Now, understand that meat eaters can get all of their essential proteins from meat, fish, milk, so from animal resources, okay? No single plant source contain all of the essential amino acids, okay? So that's why vegetarians and vegans need a combination of grains, beans, and nuts in order to get them. But it can still be obtained from the diet. It's just that they just need to combine it. It is important to note that whenever we have a peptide, we are always going to draw them from the N terminus to the C terminus. Okay, the N terminus explains that this is going to be the amino side of a peptide, and this is going to be the carboxylic acid side. Okay, now as you can see, when it comes to the different abbreviations, we can utilize the 3-letter code, but we can also utilize the 1-letter code for the amino acids. Here, specifically, as you can see, we can observe that in this peptide, we have the amino acids alanine, glycine, cysteine, tyrosine, glutamic acid, phenylalanine, cysteine, methionine, some of these are incorrect, and then we have leucine. I'm just fixing it because this PYR, there's no 3-letter code that is PRY, even if you look through the tables. But understand that when a peptide is given to you, it's going to be written from the N terminus to the C terminus. Lastly, to conclude our talk about proteins, we're going to learn about the different protein level structures. Okay, so when it comes to the protein structural levels, there are 4 protein structural levels. The first one, which is the primary structure of a protein, it is just the sequence of amino acids that are present in your protein structure. Now, understand that when it comes to this, it is very important that here in the slide we have the structure of insulin. Insulin, as you can see, it is 2 chains that are linked together through these red bonds that we have here, and I'm highlighting in green because we know that cysteine residues, okay, have an SH bond. One of the things that cysteine can do is that they can make a disulfide bond. So when that cysteine residue is oxidized, one of the things that we observe is that it results in a disulfide bond. So I just wanted to illustrate it. But again, the primary structure of a protein is the sequences of amino acids that it has. The next level, which is the secondary level for protein structure, understand that this is going to be the 3D shape of a localized regions of a protein. Now, specifically, when it comes to this is going to be largely dictated by hydrogen bonding interaction along the peptide backbone. What this means is that the interactions that are going to be occurring is between the oxygen in the carbonyl in the peptide and the hydrogen of an NH group down the road. Okay, so it's not going to be the ones that are neighbored, but just imagine that this hydrogen from a residue more down from that oxygen is the one that they're going to be interacting. Now, when it comes to the secondary structure, there are 2 common arrangements for them. One is going to be the alpha helix, which is going to make this helical structure that we have here. And then we're going to have the beta pleated sheet. And this beta pleated sheet, as you can see, it is illustrated down here. it is important to note that the hydrogen bonding, as I previously mentioned, is happening between the backbone, meaning it is happening at the peptide bond. The side chains are not involved in the hydrogen bonding for the secondary structures. Understand that what makes a sequence of amino acids adopt a alpha helix or a beta pleated sheet is going to be something that is called in biochemistry the secondary structure propensities. But that is something that we're not going to be going into detail in this course. The next protein structural level is the tertiary structure. And the tertiary structure of a protein refers to the 3D shape of the entire peptide chain. Understand that the shape will change depending on the solvent and the environment, but one of the most important things to take into consideration is that the tertiary structure results from interactions between the side chains of the amino acids. Now, understand that if you have a polar environment, the protein is going to adopt a conversation that is going to put the polar groups in the exterior. If you have a non-polar environment, the polar groups are going to be in the interior. But for the most part, what we observed is that most of the time our proteins are going to be living in a polar environment, because remember that in the body, the most common solvent is water. One of the things that can occur to proteins is denaturation. And understand that denaturation is going to be different treatments that can happen to a protein, and then is going to create unfolding. Things that affect protein folding, and it's going to be affecting the intramolecular forces that are holding that protein, include temperature, pH, agitation, the use of acids and bases, and even the use of organic solvents. The last protein structural level is the quaternary structure. Understand that the quaternary structure is not present in all proteins. The primary, secondary, tertiary is present in all proteins. Now, the reason is because the quaternary structure is specifically found in proteins that have different subunits that have to come together in order to make the final structure. To give you guys an example, here we have the structure of hemoglobin. Hemoglobin is made out of 4 peptide chains, and they have to come together to make the protein of hemoglobin. So unless your protein has subunits, that's the only time that is going to have a quaternary structure.